if you sit there at the panel table and Renee can join you, I think what we're going to do is just add people as we go. So we're going to try to keep this to 10 minutes or less so that we can catch up from where we were behind this morning. Anybody want to query Ben? Alta.
No,
Oh,
no,
you're
I'm
raising your hand, or you're
change.
scratching your head.
I'm thinking.
Yeah. So I mean, part of the I'll talk while others think. It's always dangerous, but I'll do it. So it seems to me part of the lesson from pediatric gene transfer is it's a case where there's lots of first time in humans and first time in pediatric humans. And the RAC has had to wrestle with that in a very much ongoing way. So you talked kind of around that, but you didn't really sort of focus on how to think that through from an ethics perspective.
You know, what's interesting about that is I think that there was a lot of concern. I mean, I'm thinking of these CF trials, which were actually not the original trials, but the later ones that were in adolescents. Huge amount of concern about risks, because it was not only the risk of the procedure, but it was also the risk of the bronchoscopy, the risk of the sedation. This would be on placebos. So, you know, really some complicated issues. And what I would say that they never really resolved those issues, other than relying on this very intensive approach to permission and consent. And so I think that was actually used as a rationale for going forward, rather than wrestling to ground the issue of is this safe enough. Although I believe there was an underlying assumption that this could be done relatively safely, but all those issues were still there.
Yeah. See.
Either in the preclinical studies leading up to gene transfer experiments or during the clinical trials, could you comment a little bit on to what extent unintended modification of the germline was a consideration? Is that something that's included in a consent? Is that something that the FDA requires some preclinical safety information on?
I'm not sure I can answer that definitively, so I don't want to say anything incorrect. What I would say is that because actually I did a project where we looked at over 400 consent forms, and which is now over 10 years ago, so I can't recall the details of all that. My recollection is that some of those did mention that they talked about a wide range of risks, including unknown risks, and though as well as that specifically. Again, one of the challenges of consent forms is that these consent forms are incredibly long, and one of the biggest issues with the consent forms for gene transfer research is that they really often, while they were clear in the benefit statement, often to say this may or may not benefit you, but then the purpose, they would go on to explain what all the benefits are. I think the whole consent process and the forms are incredibly complicated and very challenging, and I don't think we'll solve problems by simply having one extra line on the form. I think the question is deciding how critical is this issue, and is this one of the 5 or 7 issues that people need to think about ahead of time? It probably is, and then we need to figure out how to address that effectively.
Alta.
A few people now have talked about the importance of long-term follow-up studies and longitudinal studies and some of the challenges with them. When you were working on these protocols, what arrangements were you making, or were they making for long-term studies of the stability of the genetic change, the stability after the transfer has been made, as well as for the kind of, presentation in terms of health? I mean, both the more...
I can comment in 2 different ways, and I think this is actually a challenging issue. This is not my field of expertise. I have a fair amount of anecdotal experience. One of the things that's relevant for the children involved in the initial ADA SCIDS treatment is that, which was done at the NIH Clinical Center, is that there was long-term follow-up of those children, precisely because of the structure there and the opportunity for long-term support. The gene transfer trials for cystic fibrosis I was developing were done by a pharmaceutical company, which no longer exists now, and the building is now owned by Seattle Children's. And so one of the challenges is that, the requirement for the long-term follow-up requires an entity that's still going to exist, and we don't know whether the entities who are sponsoring the trials will always be existing. So that does create problems and requires perhaps mechanisms other than the sponsor to be responsible for making sure that those studies are conducted.
maybe we catch up a few more minutes then, unless others have other thoughts. Great. Thank you, Ben. So that